in the Soil, Water, and Ecosystem Sciences Department. She is co-advised by me and Dr. Ashley Smith at the Tropical Education and Research Center. Before coming to UF, Alex did a bachelor's and a master's degree at the University of Virginia. While she was doing her degrees in Virginia, she was actually doing her research still here in Florida. She's one of the few people who have looked at the genetics of Syringodium filiforme or manatee grass. So she's been working in Florida for a while. In between her master's and her PhD, Alex moved to Washington, D.C., where she worked for the US EPA as an ORISE fellow. And I think that really sort of defines a little bit about Alex as a scientist. She thinks about problems that are really important, things like water quality, carbon cycling, which you'll hear some about. And she thinks about and does her work in a way that there's real application. So that's really cool. And I think something that defines her as a scientist. The work that she will present today will be an example of that. She's going to present some really cool data that is important for a number of reasons. First, it really pushes the boundaries of ecological theory. It has real applicability. She's going to talk about carbon cycling, and those of us interested in global climate change really need to think about carbon cycling in this way. It also is really important because it highlights the importance of monitoring. So UF and DEP are investing a lot of resources in monitoring in and around the Nature Coast Aquatic Preserve, and Alex's work is going to show us why that's important and the things that we can do with that monitoring data. So I will turn it over to Alex.
Thank you for that. Today, I'm going to talk about drivers of carbon storage and carbon loss through greenhouse gas emissions in Cedar Key seagrass meadows. And the title says to think small because we're going to be looking at differences in storage and emissions at a relatively small spatial scale. I'll give a little bit of an introduction, even though most of this audience is familiar with seagrasses and all the wonderful ecosystem services they provide. Seagrasses are marine plants that line the coast, often forming extensive meadows like the ones we find along the Nature Coast. They're foundation species that provide habitat for commercially and recreationally important fish and shellfish like scallops, and they're eaten by iconic sea turtles and manatees. They can provide some level of coastal protection as a living shoreline, and they can improve local water quality. And recently, they've received a lot of attention for their ability to capture and store organic carbon in their biomass and underlying sediments, aka blue carbon. While we're still researching how well seagrasses can store carbon, a lot of groups are seriously considering or already implementing the preservation and restoration of seagrasses to lock away carbon and mitigate climate change. For example, the Indonesian government is creating policy to protect seagrasses for this purpose, and the first voluntary carbon credit system offering credits for seagrass in the US has launched recently in the Chesapeake Bay. And so while it's exciting that coastal ecosystems can provide this benefit of carbon storage in addition to many other ecosystem services, some researchers are concerned that the benefit of carbon storage is a bit overstated, mainly because, well, the carbon cycle is a bit complicated. So the diagram here may be a bit of an oversimplification of carbon cycling, but it highlights our assumptions of how blue carbon works. First off, seagrasses and other types of coastal vegetation like mangroves and salt marshes, they're all plants, so they consume carbon dioxide during photosynthesis and can convert or fix some of that carbon into biomass. So in that sense, they're removing some carbon from the atmosphere, but the amount of carbon stored in this way pales in comparison to the amount in the sediment compartment. The most impressive pathway of carbon storage in coastal systems, especially seagrass meadows, is the accumulation of organic carbon in the underlying oxygen poor sediments that can be where organic carbon can be preserved for millennia. And if this organic carbon were not buried under these ecosystems, it might be available for organisms to break down and respire back to the atmosphere as carbon dioxide. But these ecosystems do naturally produce carbon dioxide and other more potent gases like methane. And in order for blue carbon strategies to work, the assumption that only some carbon returns to the atmosphere needs to hold. So whether these ecosystems store more carbon than they release is a motivating question for my research. And I'm specifically looking at whether greenhouse gas emissions discount storage in seagrass meadows. I'll provide a little bit more background on how carbon is stored and emitted from seagrass meadows. And like I said, the most important contribution seagrasses make to blue carbon is trapping carbon in sediments. The organic carbon that seagrasses store can come from a variety of sources, including the seagrass itself as it decays, benthic algae or phytoplankton, in our area, marsh and mangrove from nearby litter, intertidal habitats, as well as riverine derived particulate or dissolved organic matter. So seagrasses are particularly effective carbon sinks because they can intercept this flow of organic carbon and immobilize it within the meadow. The seagrass canopies can slow wave energy and currents and allow particles to settle on the sediment surface. And then at the sediment surface, seagrasses promote long-term storage by continuously accreting and accumulating sediment and eventually burying organic matter in oxygen-poor sediments that prevent decomposition. The seagrass rhizome and root system stabilizes those sediments and prevents resuspension of the deposited organic matter. And seagrasses have a widespread geographic distribution. So that means they store a lot of carbon and actually play an important role in the global carbon budget. So there's a lot of interest in how we might manage and restore meadows as natural tools for climate change mitigation. But we've begun to question whether carbon stored in seagrass sediments outweighs carbon loss through greenhouse gas emissions, which include carbon dioxide, but also methane and nitrous oxide. And these gases are part of natural processes occurring within seagrass meadows. Carbon dioxide is consumed during photosynthesis and produced during respiration. Methane and nitrous oxide are produced and consumed mainly by microbes within the sediment, but not the plants themselves. And methane and nitrous oxide are especially important to study because they have very high atmospheric warming potentials. Methane has a warming potential of about 30 and nitrous oxide has a warming potential of about 250, meaning that within a 100-year period, 1 ton of methane emissions will have a 30-fold global warming impact than 1 ton of carbon dioxide emissions. So due to the high impact of these gases, it's really important for us to quantify them for different types of seagrass meadows so we can learn what controls their production. So the focus of my talk is understanding what tips this balance between storage and emissions at small spatial scales in a local seagrass meadow. And I'll first present our work on carbon storage. So we can't properly manage and restore meadows as part of emissions reduction strategies unless we understand what drives variation in carbon, especially in multi-species meadows typical of tropical and subtropical regions like the Nature Coast, which are somewhat underrepresented in carbon stock estimates. The Forkran et al. Figure here highlights variability in seagrass carbon storage globally. The figure shows carbon storage by ecosystem type, and it shows the amount stored in living biomass versus the soil. Seagrasses are much smaller than mangroves and trees, so they store much less carbon in the above ground component. And I just want to highlight with this figure that seagrass carbon storage in the soil is highly variable globally. They can store a small amount of carbon or a high amount of carbon shown by the spread of these data points. And other studies have actually found similarly high variation regionally throughout estuaries and even within meadows. So to better understand the small scale drivers of variation in storage, we investigated the effects of historical and contemporary seagrass characteristics on meadow carbon stocks. We conducted our work in Cedar Key, where like the rest of the Gulf of Mexico, tropical species often coexist, and those include Halophila engelmannii, Haliduli wrightii, Syringodium filiforme, and Thalassia testudinum. And I want to emphasize that Halophila and Haliduli are smaller and have higher growth rates, while Syringodium and Thalassia are larger and have slower growth rates. And I mention this because these traits may be important to carbon storage at the meadow scale. So in a relatively small area in Cedar Key, we have a variety of species compositions to help us determine whether species plays a role in carbon storage. We also conveniently have a trove of publicly available seagrass monitoring data from the Florida Department of Environmental Protection. So we can test whether historical seagrass meadow properties matter to carbon storage now. So I'm excited about this because most studies researching seagrass effects on carbon measure contemporary cover, contemporary biomass, or other contemporary properties, but we were able to use these historical data sets to generate additional predictor variables and examine the role of site history in carbon storage. So here are hypotheses for our research questions on the small scale drivers of carbon storage. First, we expected seagrass species diversity, both contemporary and historical, to enhance carbon storage, because diversity in general can promote ecological processes and functions, but more specifically, we suspected having more species present adds complexity to above ground and below ground structures. And maybe this complexity enhances seagrass carbon trapping by the canopy and burial and stabilization within the rhizosphere. We expected historical cover to predict carbon storage because storage represents accumulation and burial over a long time. So in this bottom figure, consistent levels of cover year to year should lead to higher carbon storage because there are plants continuously present to trap and bury carbon. So to test the effects of meadow characteristics on carbon storage, we paired field sampling with analysis of long-term monitoring data sets. We calculated a number of variables from the historical data sets that might explain carbon storage, like historical cover, variability and cover over time, historical species diversity, and relative dominance by each species. Then we returned to the 25 Florida Department of Environmental Protection monitoring stations in Cedar Key and measured sediment carbon stocks. We estimated carbon stocks, which is the mass of carbon spread over an area, by using percent total organic carbon and dry bulk density. We focused on surface sediments because surface sediments likely accumulated more recently, which better matches the timeframe we have monitoring data for. While we were sampling carbon stocks, we also measured contemporary seagrass biomass, species diversity, and relative dominance by species as potential contemporary drivers of carbon storage. And back in the office, we also collected some data on environmental variables that might influence carbon storage. And I'll share those results first. So we found that common environmental drivers of carbon storage did not have strong effects in Cedar Key. We looked at water depth because that can control how likely a site will be a depositional environment where carbon can accumulate. And most sites were around a meter in depth, and the small variation in depth that there was across the sites did not really affect carbon stocks. We also looked at distance to navigational channels as a proxy for disturbance, maybe caused by boat scarring. And here I show the main navigational channels recreational boaters might use around Cedar Key in orange. So we measured the shortest Euclidean distance between the sites and the channels using actual mapping software rather than my hand-drawn diagram here. But distance to channels also did not affect carbon stocks. But I do have a feeling many people do not restrict themselves to marked channels, so maybe this is not the best proxy for human-caused disturbance. We also looked at relative exposure using the relative exposure index, which considers wind velocity and direction and site distance to land. And it's an indirect measurement of wave energy, but it also did not affect carbon stocks. So the fact that these broader controls on carbon storage were not so important here builds a case for examining more localized controls on carbon storage, like site-specific meadow characteristics. But even if the environmental variables we chose to look at didn't influence carbon stocks in Cedar Key, we also wanted to make sure there wasn't some underlying environmental gradient that we didn't measure that controls carbon storage. So the figure here is a map of our 25 sites colored by carbon stock quantiles. White is the lowest carbon storage and red represents the highest carbon storage. And visually, we can see the low carbon sites and high carbon sites are found throughout the area in a seemingly random pattern. We also assessed this more quantitatively and did not find evidence for spatial autocorrelation. So this allows us to focus on the other drivers of carbon storage that we're interested in. And we did find that site-specific drivers affected carbon storage. In this and the following figures, carbon storage or stocks are represented as milligrams organic carbon per cubic centimeter on the y-axis, with explanatory variables on the x-axis and associated linear regression equations and stats above the plot. Remember that explanatory variables from the monitoring data set are referred to as historical, while explanatory variables from the newly collected data are referred to as contemporary. So the figure on the left here shows there's a weak relationship between contemporary species richness and carbon stocks. And we took the log just to conform to some statistical assumptions, but we expected a complex canopy and or rhizosphere in terms of leaf and rhizome morphology might promote carbon storage through enhanced trapping and stabilization. And this might be true, but the R-squared value here is on the smaller side. So a lot of variation in carbon storage exists across these sites and is left unexplained by contemporary species richness alone. The figure on the right shows that carbon stocks increase when the relative dominance of Thalassia increases. Put another way, meadows dominated by Thalassia, the larger, slower growing seagrass, stored more carbon than meadows without Thalassia. Our findings did somewhat support our hypothesis that more diverse meadows would have higher sediment organic carbon, but clearly species identity is an important part of the story. Historical species identity and richness also predicted sediment organic carbon. The figure on the left shows the relationship between historical species richness and carbon storage, and this is a positive relationship. Historical richness here was calculated as mean species richness over the entire monitoring period, and the relationship is slightly stronger than it was for the contemporary species richness. And maybe having a higher number of species over time is more important than diversity at the time of sampling stocks. The figure on the right shows the relationship between the historical relative dominance of haloduli and carbon storage, and it's a negative relationship. And maybe that's because haloduli tends to dominate in more marginal or stressful environments that are prone to sediment erosion and erosion of carbon stocks. So these results are also hinting that diversity may be less important than species identity in driving carbon storage. And so using structural equation modeling, we found strongest support for our hypothesis that historical cover and variability in cover drives carbon storage. But here I'm still showing the individual linear regressions because they're a bit easier to interpret. So for the figure on the left, the explanatory variable is historical cover. It was estimated using Braun-Blanquet, a proxy for seagrass cover. And I know the Florida Department of Environmental Protection uses a slightly modified scale, but I don't think this affects our analyses very much because we're most concerned about relative amounts of cover and changes in cover rather than the precise percent cover. So we see that as historical cover increases, carbon stocks also increase. We also found that consistency in cover promoted greater carbon stocks. For the figure on the right, the explanatory variable is the coefficient of variation of the Braun-Blanquet observations over the entire monitoring period. So as variation in cover increases, carbon stocks decrease. We can also think of this in terms of stability. With less variation in cover, there's greater meadow stability, which promotes greater carbon storage. So to sum up our findings from our carbon storage work in Cedar Key, we found that consistent historical cover or stability promoted carbon storage, while contemporary drivers were less important. And species identity, namely the presence of long-lived species like Thalassia, also promoted storage. This may be because Thalassia tends to grow in more stable environments, but Thalassia can also promote more stable sediment conditions with its larger rhizomes. And a recent global meta-analysis of seagrass carbon storage found that meadows dominated by long-lived species tend to store more carbon across the globe. And our findings suggest that this may be because long-lived species are potentially indicators of more stable conditions that promote carbon accumulation and burial. I also want to emphasize the importance of long-term monitoring. The Florida Department of Environmental Protection datasets allowed us to ask these questions about historical drivers of seagrass carbon storage, which is usually not possible. And just to step back a minute and think about these results within the broader framework of carbon storage versus losses, we found Cedar Key meadows store carbon in amounts comparable to other Gulf of Mexico meadows. It's the value highlighted here. And because we were working at a relatively small spatial scale, broader environmental controls were not so important in driving carbon storage. So we were able to measure how seagrass characteristics and site history influence sediment organic carbon, and detected some potential mechanisms that may explain the extreme variation we find in seagrass meadow carbon storage globally. These mechanisms include stability in cover and species identity, especially the presence of thalassia, which both positively affected carbon storage. Now I'll talk about drivers of greenhouse gas emissions. We established from the first part of the talk that seagrass meadow carbon stocks are highly variable globally. We also know that seagrass meadow emissions are highly variable. The figure here shows the range of emissions in millimoles gas per square meter per day. And for ease of comparison, the units are in carbon dioxide equivalents. So positive values here indicate emissions or net loss to the atmosphere, while negative values indicate consumption. So there are some very negative values for carbon dioxide because seagrass meadows can be highly productive, acting as carbon dioxide sinks. Again, I'm focusing on methane and nitrous oxide though, because they have very high atmospheric warming potentials. So let's zoom in to get a better look at methane and nitrous oxide. The range of emissions reported in the literature shows that most meadows are net neutral or net emitters of methane and nitrous oxide. So if there's interest in managing meadows for carbon storage, it would be helpful to know what makes some meadows emit more greenhouse gases than others. And there's a suite of environmental factors that likely contribute, but we'd like to know if there are plant characteristics that affect emissions at small spatial scales. So as I stated before, and you probably already know, multiple seagrass species coexist along the Nature Coast and specifically in Cedar Key, the site of our study. So we have the opportunity here to explore how emissions differ by species living under the same conditions. And the two most common species we find in Cedar Key are Haloduli wrightii. Remember, this is the smaller, faster growing seagrass, and Thalassia testudinum, the larger, slower growing seagrass. So based on some physical differences, we might expect seagrass species to affect the magnitude of meadow greenhouse gas emissions. Here are hypotheses for our research question of what drives emissions at small spatial scales. First, we expected vegetated sediments to produce more greenhouse gases because vegetated sediments can concentrate organic matter, fueling microbial activity, the main pathway of greenhouse gas production in these environments. Second, we expected higher emissions in warmer months because both plant and microbial metabolism increase with temperature. So with higher temperatures, maybe higher seagrass biomass increases organic matter levels in the rhizosphere, fueling even more microbial activity. Third, we expected to find differences in the magnitude of emissions between meadows dominated by different seagrass species for a number of reasons. But for example, different species may have different capacities to oxygenate and release carbon compounds into the rhizosphere. And we figured that a more oxygenated and carbon-rich rhizosphere would enhance microbial activity and promote greenhouse gas production. So while we expected emissions to differ between the species, we weren't sure how exactly they would differ. To test our hypotheses, we measured sediment water fluxes using both dark and light incubations. We did this because different metabolic processes dominate under dark versus light conditions, like plant respiration and photosynthesis, for example. We sampled across seasons and 4 vegetation treatments, including an unvegetated sediment or a bare control, and sediments dominated by Haloduli or Thalassia, or in a mixed treatment with both species present. I'd like to pause here to mention that I've been talking about emissions as gas loss to the atmosphere, but we measured sediment water fluxes in our study because that's where the gases originate, and that's where we can better understand the mechanisms controlling greenhouse gas production. But we're ultimately concerned about loss to the atmosphere in terms of using carbon storage as a hedge against climate change. So for each seasonal incubation, we collected cores from a meadow with both species growing intermixed and side by side, and from some adjacent bare patches. We collected 6 cores per vegetation type. And as an example, on the top right, that's an August core. It has lots of green shoots, but the bottom right is a January core, and it's very sparse. So we brought the cores back to the lab where we ran the incubations. The cores were close to the water bath, and we took water samples at regular intervals so that we could estimate gas production and consumption. And we did this under both the dark and light conditions using grow lamps for the light conditions. So once we collected a water sample, we used the headspace equilibration method to measure dissolved gas concentrations. And this method involves a lot of shaking. And it basically moves the dissolved gases in the water sample into a headspace so that gases in water and air are in equilibrium. And then we can measure the gas concentration of the air sample on a gas chromatograph, and then use solubility equations and gas laws to back calculate how much of that gas was dissolved in the water sample. But all you need to know is that we calculated daily fluxes based on the sum of dark and light fluxes, assuming a 12-hour dark and 12-hour light cycle. So here are the summed methane and nitrous oxide fluxes as carbon dioxide equivalents across all treatments. So to orient you to this figure and the next few figures, the x-axis is the treatment starting with the bare sediment on the left, followed by haliduli dominated sediments, the mixed treatment, and thalassia dominated sediments on the right. And the y-axis is the gas flux, which here is millimoles gas per square meter per day. The sampling season shown by color as May, August to January from left to right. And p-values for Kruskal-Wallis tests on main effects, vegetation type, and season are shown by the core and thermometer symbols in the bottom right. So the figure indicates total fluxes were highest in vegetated sediments. And that's the main vegetation treatment difference we detected just between the bare sediments and vegetated sediments. There was no apparent difference between haliduli dominant, thalassia dominant, or mixed treatments. And January fluxes were generally negligible across all treatments. So there was some level of seasonal effect detected here. And this could be because water temperatures were cooler, microbial activity was lower, and seagrass biomass and metabolism were also probably lower in January. Now I'll show you how each individual gas contributes to this pattern. And nitrous oxide did not contribute very much. Net nitrous oxide fluxes were small, close to 0. So if you look on the y-axis here, the units are in micromoles and the numbers are pretty small. But methane fluxes were net positive in vegetated sediments, and this is what was driving the overall pattern of higher total fluxes in vegetated sediments. There were essentially no fluxes from bare sediments, and there were, again, some seasonal differences where January fluxes were negligible and fluxes were highest and most variable in August. We suspect these patterns were driven by temperature and changes in seagrass biomass. Again, no clear difference between the species categories here. But now I'll show you how seagrass biomass correlates to methane fluxes. In this figure, I show the relationship between seagrass biomass on the x-axis and methane fluxes, again, on the y-axis. And the figure shows that biomass and fluxes are positively related, but maybe weakly. We can look more closely by species with haliduli in gold, thalassia in green, and the mixed treatment in gray. We still see a similar pattern across species where biomass drives fluxes. And we also found similar patterns for other plant traits, like the ratio of above ground to below ground biomass, total leaf area, and total root area. To recap, we found support for our hypothesis that vegetated sediments produce greater fluxes than bare sediments. We also found support for our hypothesis that fluxes would be greater in warmer seasons, but fluxes did not vary by species. So to put our data into context, we found that fluxes from vegetated sediments fell within the global range of reported values. And here I added the mean fluxes across seasons from vegetated treatments on top of the figure showing the global ranges. So remember that our nitrous oxide fluxes were near zero, but our methane fluxes were net positive. So while this data tells us that methane's coming out of the vegetated sediments and into the water column, the amount overall is still pretty low. So to conclude, we found these meadows store carbon in amounts comparable to other meadows in the Gulf of Mexico, and that stability in cover and species identity, namely the presence of long-lived species, did affect carbon storage, but broad-scale environmental drivers were not so important. We identified that for at least part of the year, Cedar Key seagrass meadows may be net emitters of methane. We found that the magnitude of these emissions differ by season and biomass, but not by species. And just a quick note here that our comparison of storage and fluxes is not directly comparable because the units are slightly different. So the jury is still out on the bigger question of whether these meadows in particular or meadows more generally are carbon sinks. But we found an important pattern where meadows with Thalassia testudinum stored more carbon, but meadows without Thalassia testudinum did not necessarily, or with Thalassia did not necessarily produce higher emissions. So that's good news if we're interested in managing meadows to mitigate climate change. With that, I'll thank everyone who's helped with this project, and I will take any questions.
If anyone has questions, please enter them in the poll, or you're also welcome to unmute yourself and ask Alex questions directly. While people are thinking, I can go ahead and get started. You talked a little bit about the importance of species diversity and saw some potential impacts for species diversity, and you found it in some cases, but not others. But when you did find them, it was positive. Are there any other ways that species diversity might impact ecosystem function of seagrasses? Are there other negative reasons? Are there other negative implications of diversity?
Not that I can think of. I typically think of the positive implications of biodiversity. Yeah, I think sometimes more diverse sites can be associated with more recently disturbed sites, but that also can just be part of this successional process of, different species having different capabilities to adapt.
Brad has his hand up, so you can go ahead, Brad.
Hey, Alex, great talk. I mean, I really enjoyed it. I think these are super important topics to be considering, especially as all the coastal states are watching Virginia very closely. You know, I know that a lot of money is being spent on clam seagrass co-planting. So I wonder what your thoughts are about the role of infaunal species in either the emission side or the burial side, which is most important would be a good place to start.
Ooh, that's a really good question. So there's definitely a lot of benefits to co-restoration and a lot of benefits that bivalves impart to seagrasses, but in the carbon realm, I would think that especially bivalves, their burrowing would kind of disrupt the sediments and then introduce oxygen and oxidize that stored carbon. So yeah, I don't think they would necessarily benefit carbon storage. That's a good question though.
Very interesting. So there are going to be lots of potential study sites available for you to look at that problem in the near future on both sides of the coast.
Totally. Yes.
Keep that in mind.
For sure.
Savannah has a question too.
Sure. Yeah. So I wonder if in your reading or discussions with other researchers or just maybe even your own work, because this was done in Cedar Key, which is kind of more of a high latitude site in terms of the range of Thalassia. I wonder how you think this may interact with latitude and potentially like biotic factors that may vary with latitude, like grazing.
That's also a really good question. Yeah, I guess we were expecting these tropical meadows at these lower latitudes to have higher emissions just because it's warmer for more parts of the year. And then the amount of nutrients and organic matter available probably really matters as well. But grazing, I think, can be just like the clams and the bivalves, it can be kind of disruptive, where depending on the grazer, they can uproot some of the grasses and they can cause some resuspension of the sediments that would probably oxidize some of the stored carbon, at least at the surface. And then longer term, if grazing has more negative impacts on grasses, if the grasses aren't there, they can't store the carbon. But I think, yeah, there's still a lot of work to do on understanding how grazing affects seagrasses in general. So that's a good question.
Sure. Yeah. So just quickly to repeat back so I make sure I understand, we might hypothesize that the higher latitude Thalassia might be more storing more carbon relative to its tropical counterparts based on temperature.
Yeah, I could buy that. Yeah, especially if they
Okay.
have
Requires more looking into, but yeah. Okay, thanks.
Mhm.
Anybody else have questions? I have one more. Alex, you point out that a lot of carbon storage stories in the literature don't look at both storage and emissions together, which you have done. But are there other things that, if we want to understand the whole implications of seagrass on the carbon cycle, are there other things that we need to be measuring as well?
Yeah, so it's not just storage and greenhouse gas emissions that matter. There's also different inorganic processes happening in the water column, like calcification and dissolution. And contrary to what you would think, when organisms calcify their shells, it actually produces carbon dioxide. So it's kind of emitting carbon dioxide. So that has been kind of brought to the forefront as another kind of big question in how seagrass meadows and coastal ecosystems actually store carbon.
So it sounds like there's a lot of work to do, thinking about bioturbation, about grazing, about calcification too. So
Yeah.
complicated issue.
Yeah.
Anybody else have any questions? If not, I think we can end this seminar, and thank you, Alex, for presentation, and look forward to seeing you all next month when we learn about prop scars from Teresa Groninger.